DNA is really the genetic treasure of the cell. DNA is the primary heritable information in living systems. It is the primary source of information that we pass down to our children. And even though DNA exists in these complicated forms, nature has provided us with tools to access those forms. In the end, like everything in the natural world, it's all just chemistry.
Science is making huge leaps.
Changing the future of biological research.
CRISPR can edit genetic mistakes.
CRISPR began by investigating how bacteria survive infection from viruses. We think of bacteria as threats to human health, as sources of infection, but bacteria are also prone to being infected. Scientists discovered that naturally occurring CRISPR is really a bacterial immune system that uses past encounters with viruses to program a set of molecular scissors, now more famously called CRISPR-Cas9, to cut DNA. It makes what's called a double-stranded break, and in the process destroys the ability of that DNA to function.
DNA are long stretches of chemical code comprised of A's, C's, G's, and T's. And these 4 letters spell out everything that's required for making hundreds of genes, thousands of proteins. And these different letters base pair or hydrogen bond with each other to functionally protect the DNA. And if one of those letters is misspelled, then the instructions which describe the proteins that are essential to life are not correct. And it turns out a lot of times diseases are caused simply by a single letter misspelling in your genetic code.
The pivotal moment in its use as a genome editing agent came in a series of papers published at the end of 2012 and early in 2013 by a variety of scientists, including Emmanuelle Charpentier, Jennifer Doudna, George Church, Fun Zhan. These papers showed that you could program the molecular scissors to disrupt the function of the genes that you're cutting. But in most cases, you may not be interested in simply destroying the function of a gene, but instead in actually repairing the gene from a form that causes a disease back to the healthy form.
CRISPR 2.0 base editing is able to affect changes that we want in the genome in a really precise manner.
Base editors are molecular machines that cleanly convert one base into another without cutting the DNA.
By binding to DNA and not creating a double-stranded break, we're able to precisely make a single-point mutation without any undesired outcomes that you usually get with double-stranded breaks. So you don't get really large deletions, you don't get rearrangements.
So CRISPR 1.0 is using scissors and glue, and CRISPR 2.0 is using a pencil and eraser.
So to develop base editing, we began by disabling the CRISPR scissors so that they no longer could make double-stranded cuts in DNA. Instead, we preserved the ability of CRISPR to bind to a target DNA sequence. That's really a very special feature of CRISPR-Cas9 and related CRISPR proteins, namely that they can be programmed with a short piece of RNA called a guide RNA to home in on just one DNA sequence of interest and not be distracted by the billions of other DNA sequences that might be present in the genome. But because DNA is double-stranded, when you change one base of a base pair to something else, you've actually created mismatched DNA. So we worried about this disagreement because the cell, in the process of resolving this disagreement, can do so either by changing the unedited strand, replacing that, or what we didn't want is it could instead replace the DNA strand that we just edited, the strand that we worked so hard to perform the chemistry on. So a very talented postdoc, Alexis Comor, took on the challenge and started to think about ways of subverting the cell's DNA repair response so that it wouldn't undo the work that our base editors were doing before we had a chance to make the change permanent. And she imagined that perhaps if we made a small change to the unedited strand, the one we wanted replaced, nicking that strand causes the cell to look at this disagreement and referee the disagreement in favor of what we just edited. It was one of those moments where you hear a proposed solution and you sort of stand in awe of how beautiful the solution is, how the natural world occasionally fits together in just the right way that makes sense and is simple and easy rather than hard and complex, even though the problems you're trying to tackle at the outset can seem almost insurmountable. To be clear, there's a lot of hard work that still has to be done, but we're incredibly excited by the fact that there are human genetic diseases caused by point mutations that might be treatable and perhaps even curable by using base editors.